thanks to all organizer for giving me the opportunity to present my work. It's my great pleasure to attend this workshop and also to share with you a modern approach try to describe large-scale cortical dynamics based on structural connectivity. So let's start. So nowadays, with the advancing recording technologies, we are able to map out the projection strength from one area to another area and at a scale of brain-wide. So for the mouse brain, although we cannot get a single cell full brain connectome, we are able to get a map of connectivity at the mesoscale. Shown here is a mouse connectome at the mesoscale data by Allen Institute. Wide field imaging technologies will help us to monitor simultaneously a large population of neurons across multiple cortical areas. And from this video, you can see there's a very rich spatial temporal dynamical pattern across the cortex. At a single chart, you can see correlated activity are distributed across multiple brain areas, which seems contradictory to our common belief that each cortical area has a particular function and those areas are decorrelated. But what we see here is they are kind of very correlated and the dynamics are highly distributed. If we do a dimension reduction analysis such as PCA, we can see usually this large scale dynamics revealed by wide field imaging is playing very low dimensional patterns. Usually only 7 to 8 dimensions are sufficient to capture the majority of variance. But the problem is if we only use statistical method, so that the top of PC we infer from PC not very stable. Their spatial patterns vary session by session and also vary by different behavior and the individual minds. So the question is, given those observations, can we understand the organizing principle of distributed cortical-wide dynamics across the mouse cortex? And also, what's the logic underlying the low-dimensional structure of cortical dynamics? And finally, to what extent the connectome itself can determine the mesoscale functional dynamics of a mammalian brain beyond a fly or silicon? So to answer those questions, here I will present a dynamic ecosystem model that integrate a data set of wide field imaging and also anatomical connectivity. So it can be decomposed to 4 parts. First, we want to prove that the mesoscale connectome indeed can predict large-scale cortical activity. To show the relation between the mesoscale cortical dynamics and the connectome, first we need to define the model architecture that are good for describing the observation at a particular spatial scale. So first, we need to define the dynamical units we want to describe. So due to the constraints of genetic tools and the imaging methods, here in wide field imaging data, we sample the entire cortical population across all layers in each cortical area. And therefore, we just use a very simplified abstraction method to namely, we just model the average activity of one cortical area. We did not distinguish different cell types, EI populations, just a single variable. And next, we want to define the connection between those dynamical units. In this case, there are several possibilities. We know for each cortical area, that has a recurrent local interaction that generates its own particular function, but it also receives long-range projection from other cortical area, maybe relevant for distributed coding. As I will show later in this talk, we can prove that for model of mesoscale dynamics, the long-range interaction play a dominant role. And so we can neglect the local interaction when we model mesoscale dynamics. So with those components, now we can define our simplified model architecture. Showing here is a network of interaction units where each node represents one particular cortical area. And then the edge, the connecting nodes represents the cortical connectivity. For cortical connectivity, here we use a measurement from Allen Institute, which is a mesoscale region-to-region connectivity from one cortical area to another cortical area. In particular, in this table, each element represents the average projection strength normalized from an input area to a target area. So now combine all those components, we want to build a linear connectome-based model that will allow us to predict single trial cortical activity based on connectome. So in particular, we focus on single child neurodynamics. We postulate activity in each cortical area, get a one-time series for each cortical area. Then we utilize activity of all other cortical area, extrude itself to predict activity of one area. Namely, in this case, we extrude SSPFD, but they utilize all activity from other area to make a prediction. And the prediction weights are completely fixed by the anatomical connectivity measured by Allen. So here we don't have free parameter in the model. And then in the end, we get a prediction with single channel temporal dynamics for this SSPFD. And then we use the same procedure to make a prediction for other area. Each time we use n-1 to predict one. And you apply the procedure to all area in cortex. And the result, we get a model that combines wide field imaging activity data and also anatomical data. So how is the model prediction looks like? So the model actually works super short. Here you can see a single trial model prediction showing pink lines compared to the data showing blue lines. At a single trial level, it can capture more than half of variance in the data. But now we also want to prove that the connectome, biological connectome, play an important role in this model prediction, because the goodness of a model prediction came from a trivial fact that in imaging data, according to all areas, they are highly correlated. So you can trivially make a good prediction with any type of connectivity. To disprove this null hypothesis, we generate a random connectivity which destroys the original structure of a connectome. And we test the model performance. We also generate another type of fake connectivity, which preserves the spatial dependence of original connectome, but destroying the detailed structure information. In this way, we can say if there are extra information beyond the spatial dependence in the original connectome, because we know that the areas are far away, their connection weak, close they are strong. In the imaging data, we also say if the two areas are close to each other, they have a more similar activity. So we want to say if the model of prediction came from those trivial facts. In fact, it's not. We test model with two different data sets from decision-making mice and also from spontaneous mice. And in both cases, we find that the model with true connectome has a much better performance than model with random connectivity or spatial shuffle, like spatial connectivity. Actually, for this random connectivity, the model performance is roughly close to the prediction by global activity. Namely, we can group all pixels at each time point to get a time series and make a prediction. As you can see, the performance close to random connectivity, but the connectome really shows a power in the prediction beyond the shuffle connectivity. So far, I have already showed a linear connectome-based model has a good model performance, but this is not the end of the story. So as we already know from PCA, that only 8 dimensions are sufficient to capture the large-scale dynamics. But here, in our linear model, we use n-1 area to make a prediction, where n is much larger than 8. So which means there are a lot of redundant information in the linear models. So to distill the linear-based model and understand better which area do make an important role in the prediction, we will go further to develop a dynamic ecosystem model of cortical dynamics beyond the linear model. So first, we need to quantify what's the contribution of each input region in predicting one target area. So previously, we just sum all the contribution from all other area weighted by connectome, but we did not see very clearly which area to make important contribution. Here we first compute the average activity of all input area, and then we dot product with the anatomic connectivity from all input area to each target area, showing a yellow box. And then we normalize the input strength for each target area. In this way, we can say the average normalized input from all input area to each target area. And from those from the normalized input plot, we can see two things. First, not all areas important for model prediction. Only a few areas are important. For example, for prediction of MOP, FRP is super important, but the rest are not that important. Another thing we find interesting is that for those all areas that make important contribution to predicting other area, they are constrained in a small subset. Basically, we can set a threshold and isolate a subset of area that make important role in the prediction. And we find that there are only 7 of them. We define those areas are driver area. So we think those driver areas are the key areas that to make a prediction, while the rest of area are dependent on those driver areas. So based on those observations, we make a hypothesis that the anatomic connectivity can be compressed in this biological architecture, namely 7 driver area make important contribution, send feed forward input to the rest of area, which we call dependent area. And then there's a recurrent interaction among dependent area, which generates activity in dependent area. But we can neglect the feedback from dependent area to driver area. And with this definition of a driver area and the dependent area, now we can relabel the anatomical connectivity. Basically, we group all driven area together and all driver area together, showing the with a white color as the dependent area and the red color are driver areas. And then we can see a small submatrix WR describes the recurrent anatomical interaction between among dependent area, and then F1 describes the feed forward projection from driver area to a dependent area. And notice here, both WR and F1 are fixed by anatomical connectivity, so there are no free parameter at all. So with those definition of driver and driven area, now we construct a dynamical system model describes the temporal evolution of activity over all critical area. So we use this X vector to describe at each time points the activity of all dependent area. We then also use a vector A to describe the activity of all driver areas. And with the assumption that we can remove the dependence between from dependent area to driver area, we can simplify the dynamical system model, only focus on the activity in driver area. And then with this simplification, we can get a reduced dynamical system model equation, which describe how activity in dependent area are updated based on the anatomical connectivity among dependent area and then the feed forward input from driver area to dependent area. Still, there's one unknown parameter in the dynamical system model, which is the time scales. We don't know how fast the dynamical system model operates compared to the wide field imaging sample rate, which is 30 Hertz. So we basically test various hypotheses. We start with a simplest hypothesis, namely the recurrent interaction in the dynamical system model is so slow compared to the imaging free rate, such that we can neglect recurrent iteration and only use the feed forward input to make a model prediction. in this case, we can get this simple feed-forward model, and the model performance is very bad. As you can see, the prediction over one dependent area, AUDP, using all information in a driver area. And now we try to increase the time scale to allow for one iteration where recurrent interaction loop between the two frames of imaging. In this case, you can see when we include this recurrent loop in the equation, the model performance is much better than previous case. And with this inspiration, we keep increasing the time scale to allow for one more iteration loop. And you can see even better model performance where we include two loops for recurrent interaction. And with those observations, we make a hypothesis that the dynamics actually reach transit equilibrium between two frames of wide field imaging, such that we can sum all possible recurrent interaction loops in the system. In this case, we can write down an analytical equation and sum all possible recurrent interaction loops. And in this case, we can simply write down an analytical equation that projects activity from driver area to dependent area. And the model performance is very good in this case. Overall, this approach shows an analytical equation enable us to map activity from driver area to dependent area without model fitting. And the overall model performance is very good to all dependent area. Shown here is the performance for each dependent area. The power of this approach is it's fully utilized biological structure information to make a mapping from driver area to dependent area without any fitting. So because of that, this relation is quite universal, because the connectome information is obtained from Allen Institute, which is a measurement of average amounts, which account for the individual variability. So in our model, we also found that we can easily generalize the model to different mice during the same task, but also can be easily generalized to mice with different behavior in different labs, which show the power of a biological structure in the model prediction. Furthermore, this model also tell us the low-dimensional activity in dependent area is determined by activity in driver area, namely the activity of all critical area. Basically, they are confined into a subspace that with the invading orientation determined by the biological connectivity. And because that basically tell us the low-dimensional space that we can measure by using PCA is actually originate from the space of a driver critical area. And we can use an encoding method to test that indeed the driver area space overlapped with the top 7 PCA space. So next, we also want to test whether those large-scale activity predicted by the model has any functional relevance. To test that, we utilize raw pixel data or wide field imaging, average activity in each area, and also the model predicted activity to decode animal movement during wide field imaging. And we actually find that the model predicted activity capture a lot of variance in behavior movement. Shown here is the first PCA of a motion energy of animal behavior video. And overall, we find that the model predicted activity capture a lot of variance that can be captured by the wide field imaging data in predicting behavioral movements. So interesting, it's also consistent with the previous study showing that the encoding in the encoding model of mice behavior movement, the weight space also low dimensional, only 7 dimensional, roughly sufficient to capture the encoding space, which consists of our model observation. So finally, we will want to go further to test to do a causal test of a dynamical system model to really show that the dynamical system model play an important role in the model prediction. Basically, there are two scenarios. So firstly, in the first case, cortical dynamics are truly generated by the cortical connectivity. Where in this case, dynamical system model is very good to model the large-scale dynamics. In another case, activity of all cortical area, actually they are just a copy of a shared inputs from some subcortical area. In this case, the model prediction is very trivial. To test those hypotheses, we collaborate with Tony Zeta lab to do unilateral laser inhibition of different cortical area during wide field imaging. Basically, those plots show the different location of inhibition. Due to the approximate bilateral symmetry between the two hemispheres in the wide field image data, we can use the difference of activity between two hemispheres as an approximation of activity modulation on the laser inhibition. As you can see from this figure, that during laser inhibition, there are significant decrease of activity compared to control hemisphere activity. In this way, we can use activity difference to estimate the modulation effect across all cortical area. So, and also this is also true for other inhibition location. So basically, due to the long-range cortical interaction, although we inhibit the local cortical area, in this case VSP, we can see that modulation effects are widely distributed beyond the visual area. And the model can actually capture this large-scale modulation effect. And we also do inhibition in different locations in different areas. And in general, the model can capture the spatial pattern of a modulation effect. So in summary, here I show connectome can predict large-scale cortical activity very well, and the dynamical system model of cortical dynamics can help us understand the low-dimensional structure of cortical activity, and the behavior model enable us to understand that the large-scale activity predicted by the model actually has an important functional role. And finally, we use a laser inhibition to causal test the dynamical system model. So overall, I think with a lot of recording technology, it's a very good time to think about brain-wide modeling. And I hope this work can provide you some inspiration for the future model building of a large-scale model. With that, I would like to thank Tatiana Engel's lab, our collaborator, our funding resource, and thank you for attention.